David Knaus. David is the president and founder of Apical Crop Science, an ag tech company that combines laboratory analysis, smart data management, and sustainable growing products to provide growers with the tools they need to grow high-yielding crops organically. As a successful farmer, college instructor, and professional crop advisor for over 10 years each, he offers a unique perspective into the challenges farmers face and how to use the best of academic research and laboratory analytics to drive a smart in-field decision-making that has helped growers across the country and internationally. Please welcome David.
Thanks very much, Sarah. Thank you again. Thank you, Sarah. Thank you, Tanio. Thank you, Dennis and Steve. It's great to follow you guys up. Thanks to everybody at Tanio and AEA for having me at the event today. I'm really excited to be here and really excited to share some insights that I've gained over the last 20 years of professional experience in regenerative and organic agriculture. So yeah, again, my name is David Knaus, and I'm the president and founder of Apical Crop Science. We are a laboratory integrated crop advising firm here in the Pacific Northwest. We provide assistance to growers of a multitude of different sizes, crops, and different growing strategies. And we try to help them implement regenerative and organic systems from wherever they are in their journey and help them succeed responsibly, financially, economically, and energetically. So personally, my background is primarily as a grower, was really where I got started. I learned the organic method when I was really young, growing up, growing food for a roadside stand and some other small farms and plots and so forth. And I really started to do a lot of professional experience in organic crop management in the early 2000s. By about 2010, I was recruited by a number of different local institutions to begin teaching regenerative and organic agriculture techniques for those institutions. And so I taught college for 3 different schools over the course of the past 12 years or so. And throughout that time, I've gained significant insight. And about 2016, I decided that it was really time to take the insights that I gained as a grower and also from the world of academia and bring it into a organized method of crop management and crop improvement for growers. So at Apical, yeah, we have lab methods that are designed by world-class scientists. We provide easy to interpret recommendations, seamless data delivery to growers, provide cutting-edge crop inputs that are all based around helping you to transition to organic and regenerative methods. So the laboratory was founded in 2016. And prior to that, we did about 4 years of research and development and recruited a lot of different application scientists and soil scientists and plant physiologists into the process of lining out how we wanted to approach developing regenerative and organic laboratory methods for growers. And so ultimately, we came up with our own method of plant sap analysis and implemented a multi-method suite of soil analysis tools, as well as precision water analysis for irrigation water. And we find that when implementing all 3 of these tools in coordinated fashion, you can really get to the bottom of crop problems relatively quickly and begin to implement regenerative and organic methods with success. But today I'm here to talk to you guys about the overarching theme of the new paradigm of crop nutrition. And, it's kind of a funny word, new paradigm or, paradigm, that word. And people don't really necessarily know always what that means. And so it's basically a pattern of something, a model that is implemented. And in regenerative agriculture, we're here to break the paradigm and we're here to implement a new paradigm. And so, but why, right? And the main thing that we've found over the years is that in talking with farmers and myself in my own experience as a farmer, which I could go into ad infinitum, we figured that there was really something wrong with the conventional way of doing something. And this is something that's continually echoed in my conversations with growers around the country and abroad. And typically they know something is wrong, they can't quite put their finger on it, and they're out in search of a better way. And I imagine there's probably a handful of folks, or maybe a significant number of folks in the audience today that are experiencing the same sort of conundrums. So, it's not easy, or it is kind of easy to say, okay, well, what might be able to be improved upon within the current paradigm. And so, first we have to define what the current paradigm is. Traditional soil management and soil testing, soluble NPK, regional crop plans by commercial interests, bare ground, erosion, leaching, waterways, and aquifer issues. The list goes on and on. Some of the other speakers here have touched on a little bit more of the macro scale, issues, and I'll kind of try to tie some of those macro scale issues into micro scale management practices that each one of us can implement. And so, what's missing from the current paradigm? I think the biggest thing that I see is the last one on the list, and that's, there is no exit strategy from the current paradigm at the moment, unless you start to look at regenerative agriculture. So when I say that, I mean, if you look at the current methods of crop management and crop plantings and so forth, what happens is, you're on this treadmill year over year, you're implying increasing inputs, your profits are declining, the nutritional content of the crops are declining, your customers are being more dissatisfied, the markets are shrinking, they're going international, etc. And all of that time, you're missing out on true soil health, consumer well-being, generational up development, and environmental stewardship, and so many other things. Not to mention, ROI from your soil health and from the atmosphere above your crops, and so on and so forth. So, but realistically, these are the list of things on the left-hand side are all things that we're experiencing in agriculture today if folks are not implementing regenerative agriculture systems, primarily. Declining productivity. I mean, there is some increased crop productivity in conventional systems, I will say that, but it comes at one cost or another, which I'll touch on in a minute. But ultimately, the declining crop productivity, declining soil health, increasing pest problems, resistant weeds, resistant insects, international competition, rising labor costs, escalating fertilizer costs, all of these are backing our farmers into a corner, specifically in the United States, but also internationally to one degree or another. And it significantly matters to a lot of people because, from the consumer perspective, consumers have figured out that nutritional content is declining in crops. Not only that, as Mr. Slaba very well articulated earlier this morning, that profits are also declining as well. And so when you look at these two metrics, these are the primary metrics that you would actually consider when thinking about actually growing food for people and earning a living at it. So what you actually have is this acceleration of business, economic, and consumer outcomes that are coming at all of us as growers, as farmers, as crop advisors, as consumers, as other people in the industry, and so on and so forth. And it's coming at us a lot faster than I think most of us are prepared to handle. And so if you think about where the old marketplace was, you're selling crops typically to a regional distribution center. The market might shift due to how much the production was that season. You're going to have to compete with, any sort of easy money in terms of loans and low interest rates and so on, government subsidies. A lot of these are focused on, local or regional markets, lack of direct access to markets for growers, as well as the inability to actually control your own destiny on 60 to 80% of the decision making and things that go into actually doing your job on a day-to-day basis. And so the new marketplace really is starting to take shape and is being driven by different sorts of regulations, global gap, organic certifications, direct markets to consumers. A lot of people have kind of cut through the variety of middlemen and going direct to the consumer and gaining an additional layer of profit margin through that approach. The other thing too that's really impacting many farmers is the international markets and how international markets come and go as new production facilities come online overseas, and that ties back into again different governmental subsidies. So, for instance, if you have a second or third world country investing large amounts of government subsidies into accessing an American market, you might see a major disruption in your crop production budgeting or your nutritional management of that crop over time, because there's going to be significant pressure on the market itself. So there's also people that are accessing carbon markets. There's also people that are using their data from both qualitative and quantitative methods to drive smart decision making and save money. And ultimately, the health of people, planets, and plants are the drivers of some of these new markets that folks are having access to and gaining additional monetary benefits on their farms. So when you think about, this new paradigm and where we're headed, there's a great number of stresses that are being put on each one of our operations. Overall economic, international, genetic, soil health, economic, geopolitical, environmental, fertilizer, resistant weeds, foreign competition, labor markets, fuel shortages, et cetera, et cetera. All of these are very strong pathway shocks that are being implemented without your ability to affect. And I'm here to show you ways that you can affect what you can affect in order to buffer your own system against some of these things that you can't have any control over. So before we get into some of the strategies, a couple other things to touch on. Another thing that maybe hasn't been discussed too much today is some of the rapidly developing innovations in agricultural tech. Every day I read about a new form of technology that's being touted as the great new thing for agriculture, and labor efficiencies, or planting efficiencies, or fertilizer reduction, or herbicide reduction, or et cetera, et cetera. All of the items on this list right here are going to be affecting the development of crop management systems over the course of the next 15 to 20 years, and probably even sooner. I was talking with a gentleman at a conference recently about his ability to drone map fields and then provide precision drone herbicide sprays to those fields and reduce pesticidal inputs up to 90% in those fields, saving farmers thousands of dollars on a day-to-day basis. This is not necessarily thought of as a regenerative technique, but in my mind, any technique that reduces pesticide inputs and improves overall soil health, either directly or indirectly, falls into that category. So, but that said, there's AI, there's robots of every different shape and size being developed, NDVI from satellites and drones, all kinds of precision ag tech in planting, new types of laboratory analysis protocols, all kinds of different biostimulants, and so on and so forth. And all of these are providing growers new opportunities to shift the way that they're interacting with both their own farm and the marketplace. So if you've been to, any sort of agronomy workshop in the last 15 or, 10, 15 years, you've probably seen this picture on the right-hand side. And, there may be somebody discussing how the law of the minimum is the main governing factor of plant growth and development. And, they're right to a degree. There's definitely something to be said for plant growth deficiencies and how those impact the growth and development of plants. But it goes a lot deeper than that when you actually start to look at our crop outcomes. And I would say this, that if your crop outcomes can't even come close to achieving what nature can achieve without our influence, then we're doing something wrong. And by that, I mean, if you walk outside in any sort of, natural environment, plants aren't being fertilized. They're growing on their own without any influence from man. And they're also not being primarily ravished by insects or disease, and they kind of go through their own crops life cycle and development. And so what we see oftentimes is that there's a huge blind spot in agriculture where the misapplication and misallocation of farmers' resources on their farms is causing a lot of the problems that we're seeing. And so ultimately, if you're applying too much or too little of your fertilizer and you're not being precise in these applications, you can have these primary sort of regional declines that I sort of refer to as, and this is very common from region to region, right? So, in our region here in Oregon, we used to grow, hundreds of different types of crops on large scale, and currently there's only a fraction of those still being grown, and the fraction that are being grown under very heavy chem ag systems that have a tendency to off-put externalities into the environment, watershed health, local species health, et cetera, et cetera. But ultimately, when you see, oh, well, we used to grow raspberries in this region, now we can't. We used to grow strawberries in this region, now we can't. We used to grow cucumbers in this region, now we can't. These are the sorts of conversations that are had, in the offices of farmers that I meet with on a day-to-day basis. And that loss of diversity, that loss of genetic diversity, the loss of yields, watershed, species, and ultimately human health is reversible with proper insight, proper applications, and proper materials. And so with that, I want to start discussing about, less bad news, and let's talk about how we get there, and how do we take all this, doom and gloom and oh, well, we've been doing all the wrong thing for how many years, and turn it on its head really swiftly, elegantly, so that you all can access really some nice tools. So what does a new paradigm might look like? So first of all, you're going to want reduced fertilizer costs, increased soil health, larger profits, increased quality of crop itself, reverse the genetic drift, kind of like Rick Clark was talking about this morning. It was really cool to hear some of that work that he's doing in soybean genetics, as well as family and community vibrancy. As a young boy, I would always, ride my bike around the area where I used to live, and it was always year over year you would see the farms just have more and more rusty equipment and become further into decline, less actively utilized, more grain silos abandoned, more barns in disrepair. That's what I mean when I say, community vibrancy. It's one thing to see a dilapidated barn on the side of the road. It's a whole nother one to see one that has a very vibrant farm operation running, on that land. Human health fortification, the Acres folks really do a really great job of nailing home the point about human health fortification. So I'd encourage, if you're not already interested in the Acres USA magazine and so on, that you would take a look at that. And ultimately, the long-term improvement of our species. There's a lot of different ways to look at our long-term health, and maybe that's via age of life, or life expectancy, or communicable diseases, etc. A lot of these things we think are potentially influenceable through crop management and crop health. So some prerequisites for understanding the new paradigm. If you're not interested in learning from Mother Nature, you should probably turn off the computer right now and go back to learning from soluble fertilizers. Secondarily, humility is one of the things that we are constantly being forced upon us by Mother Nature, and ability to observe and cooperate. So if you can start to get these sort of things through your head and ingrained in your body, that it's you're here to learn from nature and be a part of working with nature, instead of trying to potentially only look at nature through your own lens, right? There's different ways that you can look at nature, and I would just say that nature will really it's a fantastic partner to work with. So when we were starting the plant sap analysis laboratory, one of the things that we did in order to help ourselves learn a lot about plants and how they translocated sap and various fluids, and from the roots through the leaves and back and so forth, we tested a lot of plant leaves in their native environments. And so running plant sap analysis on native plants was a really fun project that we did for about the first 3 or 4 years of our research and development. And what we came to is a list of 10 principles that, if you understand these, you can then begin to take your operations step by step back towards a natural balance that utilizes the best of nature, and with minimal influence on your own behalf. So in order to understand these 10 principles, we had to look at literally hundreds of plant sap samples and across a lot of different species. And what we came up with is that plants in nature can thrive on low levels of nutrients from soils delivered by microbes. And I think Dennis and Steve did a great job. The work of James White and some of the others in the industry, you can't speak highly enough about the work that they're doing. Plants in nature maintain elements that are very balanced from new leaf to old leaf. This is a very common principle that we've found. When plants get more and more healthy, they have more and more elemental balance between new and old leaf. Element to element ratios within a plant have significant influence on plant growth and development. So your nitrogen to sulfur ratios, your potassium to calcium, your phosphorus to potassium, etc. Each one of these ratios, both in the soil as well as in the plant, have significant influence on how that plant expresses its own genetics in the field. Plant ratios between various nutrients have finite limits. Exceeding these limits causes plant decay and death. Crop species have target ranges and ratios of one element to another. Nutrient mobility in a plant determines where imbalances appear as excesses or deficiencies. Elements in excessive levels block out nutrient consumption of other needed nutrients. This is a very common topic, and I know that a couple of the other speakers have touched on this. At Apical, we were very, very acutely aware of this, and we are often really focused on managing plant nutrient excesses in order to elicit further crop response. Plants sequestered excessive toxins and nutrients in order to preserve their growth. This is why you see tip burn or brown leaves on the bottom of a plant, oftentimes when a plant has a one type of plant nutrient excess or another. Plant toxin excesses drive excessive consumption of other nutrients from the soil. And the larger the excess element, the greater consumption of other elements, and the faster that the soil is depleted. And so by understanding how this principle is actually working in practice, if you're familiar with base saturation percentage ratios, it's no different from that concept, but actually looking at it inside the plant on a multitude of ratios across all the plant's physiology, and primarily looking at the new leaf to the old leaf differences. So when nature operates according to the principles that I just outlined, she uses very little fertilizer and very large amounts of microbes and carbon cycling and nitrogen cycling through gas exchange from the plant to the soil and backwards. In that situation, the plants are using zero fertilization. They're relying completely upon microbes and gas exchange in order to receive all of their nutrients. And nature's able to do this with plants that have no predation. So using that as our example, we want to take this concept and translate it into agricultural systems that can access this new paradigm protocols, and then use that in order to regenerate farms, communities, environments, and so forth. And so the first process that we like to encourage amongst the growers and farms that we work with is for them to, first of all, strengthen the ability for the plant to respond to stress. And that means abiotic stress resistance. And so there's a couple other folks this morning that were talking about the recent floods in California. One week it's floods, the next week it's a frost, the third week it's a heat stress. The next time it's some sort of application problem, or there's an irrigation issue, et cetera, et cetera. You're always going to have stress in your fields, and that stress is going to be very, very correspondent to the plant health. Secondarily, you want to be able to respond to micro shocks in order to reduce that ability of that stress to impact your crop. So those micro shocks, such as water retention, microbes, soil colloid buffering, fertigation applications, foliar sprays, these are all types of plant response that you can elicit in order to respond to micro shocks. Third, last but not least, shrink the gap between the existing and the ideal. And so on all of our farms and all of our situations, we're not going to have the ideal. I still have yet to meet a farm that's got the ideal everything. They've got it figured out: the market, the management, the equipment, the year in, year out, they're breaking records of quality and tonnage and so on and so forth. We see a lot of folks that bump up into some really insane crop outcomes, and then they back off on their operations. What we see is folks that can really maintain consistency in their applications, their analysis protocols, and their management practices have the best success ratios over time. So the three pillars of the new paradigm: plant detoxification, mineral balancing, and biological augmentation. So we see these three key pivotal techniques being implemented over and over and over on successful regenerative and organic cropping systems. And at the heart of all three of those is carbon fertilization. So one of the things that I've been known to say, in various other talks and in my other discussions and so forth, is that carbon was really forgotten in the NPK revolution. And that's a very key point that a lot of us have missed. It's not about necessarily, having 5% organic matter, 7% organic matter on a loss on ignition soil analysis method. It's actually about having that carbon cycled on a day-to-day basis so that the plants can uptake carbon molecules, as well as there's carbon for a variety of purposes in the microbial community. And so if you don't have carbon fertilization practices implementing on your farm, it's also going to be very difficult to detoxify any plant nutrient excesses, balance your minerals, and also augment biology. And so cutting to the heart of all of these regenerative practices, this is why almost all of them start talking about carbon fertilization. But I would also like to take it one step further in that, again, it's not about having the loss on ignition organic matter number on a traditional soil analysis method. There's other ways of analyzing carbon in soils and plants through either direct quantitative metrics or indirect qualitative metrics that will give you a reading as to how well your plant is accessing carbon. The other thing that we've also noticed in plant growth and development is that nutrient stress precedes plant vulnerability. So the cumulative stress from multitude of nutrient imbalances creates long-term disease. And that starts out typically with protein denaturation. In other words, the plants begin to not be able to produce complete proteins. Once that begins to start, you have the secondary and tertiary effects of cellular dehydration and cell wall compromise. Once those take place, it's very simple for plants to be susceptible to insects or disease. And they can happen in any plant organ, and that's the process of decline. Each one of these processes is going to be limited by the amount of carbon that the plant can access in relation to its growth speed and development. So, in other words, if the plant doesn't have access to sufficient carbon during any stage of its plant growth and development, it's not going to be able to complete its own photosynthetic process as well as produce its own genetic capabilities. So each one of these individual plant stress points can be limited through plant proper nutrition if you include carbon, and also they are also reinforced through improper nutrition. So it's a double-edged sword. And here's a little bit of an infographic to kind of show you what we typically see when you have a restoration or loss back and forth, depending on the direction of the grower is going on that day, from a full amount of nutrients, availability due to soil conditions, availability due to plant physiology, and finally you get pinched down on your yield capabilities. But that's all being influenced by the mineral, physical, and biological influences of crop growth habitat. Any good agronomist should be able to tell you the large triangle and why that's impacting your crop management. But unfortunately, drilling down on the four limitations from genetic capability are a little bit more nuanced and require significant analysis of data to be able to tease out where exactly those individual limitations are coming into your crop. So ultimately, though, we can't afford not to implement regenerative and organic systems in the new paradigm. And these are the reasons why: declining productivity, soil health, pest problems, weeds, international competition. All of these are affecting all of our operations every single day. And so when you look at what there used to be the potential for, the old paradigm was cover crops, animal manures dealt with soil and plant health management for thousands of years, right? But since the 1960s, 1950s, they've been on this annual NPK program. Well, nowadays there's a little bit of a different type of program where you can look at allelopathy, biocontrols, plant stress tolerance, biweekly sugars, nitrogen gas cycling, NPK map balancing, as well as mineral balancing in the plant, in the soil, in the water soluble, in malic-3 or an acid extraction. And so the data sets are much more robust, as well as the tools. And so here are a few of the tools that we work with at Apical, but a lot of the other crop advising firms out there, I know AEA's products and Tanneo's products fall into a lot of these categories as well. The carbon complex conventional fertilizers is kind of the first step on your journey. Once from there, you can get into all kinds of different things: soluble carbon ferments, plant extracts, live microbes, microbial stimulators, natural pesticides, nanoparticle suspensions, micronized mineral particles, soluble organic fertilizers. The list just goes on and on now. And each one of these presents new opportunities for crop management, crop steering, influencing crop outcomes. So I hear a lot of questions in the agricultural community about soil analysis methods and protocols, why they do or don't match the crop outcomes. And I think, so in response to that, I created this slide because I do think it has some real relevance for the curious grower that's wondering what the difference is between each one of these types of soil analysis methods and which one they should use. So I think, generally speaking, the best way to look at these is their how they correspond to a different type of farming system, how they're going to respond to a different type of pest management strategy, and also how they're going to respond to crop management input types that would be used in that system. And so when you start to segment these different soil analysis methods like that, you actually have a relatively straightforward system that actually starts to make sense to the average grower. If you're in a, it's very difficult to even implement a regenerative system without at least doing a malic-3. But even folks that are trying to do a malic-3 can't reach regenerative outcomes, or definitely they won't be able to achieve organic outcomes without potentially having some problems. So oftentimes we recommend a multitude of different soil analysis methods to tease out individual data points that can impact your crop, are impacting your long-term soil health, or might be impacting your crop management plans on it from a year-to-year basis, and helping you sculpt your fertilizer analysis needs. So similarly to that, with plant analysis methods, there's a lot of different ways that these days you can analyze plants, and everything from satellites to drones to tissue to handheld types of analysis methods and so on. Each one of them has their place in the agricultural community, and each one of them has their strengths and weaknesses. And so this slide kind of illustrates some of our personal opinions on which ones have different strengths and weaknesses, and which ones you should be accessing if you're looking to have a particular sort of outcome. Obviously, we're very interested in plant sap, as well as some of the spectral imaging at Apical, and each one of those has its own benefits and strengths and weaknesses. We feel primarily that no one method of analysis is beneficial to singularly rely on. It's only through the successful application of a multitude of analysis methods that you will get repeatable outcomes that you're going to be satisfied with. So a lot of these techniques that we advise other growers to implement, such as preventing leaching through carbon applications, buffering the total volume of soils versus buffering your soil root interface. This is a key point, because a lot of times, if you do a malic-3 analysis, you'll get some crazy number. You need, 12 tons of lime or 15 tons of gypsum in order to achieve a proper calcium ratio. And that's obviously not economically or advised for a number of different reasons. But we feel that if you can just buffer the root soil interface and do it consistently, you can gain a much better control over your crop outcomes and begin to take your first steps on regenerative and organic methods. So supplementing and feeding biology, I think Steve and Dennis can speak to that significantly more than I can. But talk to those guys. They're kind of the experts in that area. But what we generally are a little bit better than those guys at potentially is foliar applications that are precise and targeted. And using those precise foliar applications can be extremely impactful, especially when you include certain ultra trace elements such as molybdenum, cobalt, certain forms of iodine, and there's others as well. So carbon as a fertilizer is a key point in this journey. And if you're not including carbon in your thinking, just stop thinking about regeneratively, because they go hand in hand. Ultimately, there's a saying we have at Apical: organic molecule synthesis creates plant-soil symbiosis. So in other words, if you have carbon-based molecules being synthesized in the soil, you're going to create a symbiotic environment where that plant and the soil can actually feed on one another and create better crop outcomes for you in the long term. Those organic molecule reactions in the soil actually are catalyzing larger scale changes within the crop's DNA, which produce higher quality yields and tonnage. If we only apply enough carbon for the crop, not the soil, the crop will build soil on its own. So a few different types of applications that can also speed up the process: prebiotics. So organic acids are a big favorite of ours. Simple sugars, other types of organic functional groups that have been maybe harvested from fermented microbes, amino acids, et cetera, et cetera. Microbial augmentation as one of the pillars of the new paradigm. And if you go to the Tanio website, or if you go to some of the literature that we provide on our website, where we drill down on which ones of the individual microbial communities you can access for specific crop benefits, there's a lot to be gained here. And folks that continue to discount the benefits of soil microbiology and maybe say, oh, well, it didn't work on my farm, I'd encourage you to try it by priming the soil and making it into a receptive environment through carbon fertilization. Not to make things too confusing, again, all the different types of crop applications that might be available to the individual grower, we like to try to segment some of these into their primary regenerative or organic or hydroponic or biological type of growing protocols, and figure that these ones have a tendency to fit a little bit better in those specific systems. It doesn't mean that they don't apply to some of the other systems, but they have a very, very strong correspondence to these other systems. So for instance, if you're doing organic crop management, you're obviously not doing any sort of carbon complex soluble nitrogen, but you will need to be doing soil building, organic suspensions, and soluble foliars. Primarily, you might be using biocontrols and plant extracts for plant disease management, because you can't use conventional IPM or even softer chemistry IPM. So once we've done some of these things, and this is kind of your homework that you need to be doing and thinking about when you're driving from place to place, or in your spare time, or when you're have some road time or whatever it is how do you synchronize where you are on your journey with some of these charts that I'm laying out here? Are you still of the mindset where carbon just raises my fertilizer bill, or microbes are bad? If that's the case, just stick with soluble NPK. But if you're starting to think, well, I heard about microbes and maybe I want to try some of these things, right? So then that kind of falls in the middle. But, if you're like me, and some of the folks that have worked with us for a while, you get into this, microbes are good. You got to maximize all your microbes. You got to maximize your carbon cycling. You got to deal with your weeds in an integrated way that's organic, that doesn't involve, harmful pesticides on the soils. So ultimately, what we see with growers that are successful or unsuccessful, it really boils down to how they're accessing the total coin. And from our perspective, these are the different pieces to the coin that growers can access that make them successful or not. Like I mentioned, so biological augmentation, carbon complex nitrogen, mineral colloid buffering, water treatment, and cation exchange balancing. Each one of these has a role to play. If you focus on one or two, you may be accessing part of the coin, but you might not be accessing the entire coin. Regenerative order of operations in the new paradigm: manage your plant nutrition to manage your biomolecules. That improves crop growth, improves crop yield, and provides economic returns. And you do that over and over and over again. In the process, if you can detox the plant and soil environment first. Second, provide abiotic stress relief. Third, adjust the carbon and nitrogen ratio of the growing crop. Fourth, balance the minerals through targeted foliar sprays or fertigation. And fifth, any other form of physiological supplementation that might be needed: cuticular waxes, protein synthesis, sugar augmentation, other forms of plant extracts that might be needed, biopesticides, microbial augmentation, endophyte supplementation. Each one of these have their place in regenerative management systems. When you do that, you'll be able to start accessing each step in the process. And as you access each step in the process, all these layers start to fall into place. The insects and disease go back to their necessary places. The biomolecule regulation induces your stress mitigation. Your nutrient regulation influences your growth management. And your abiotic stress is how you either increase or decrease that speed of that plant growth. So I'd like to just say thank you very much to the folks at Tanio and the folks at AEA for allowing me to speak today. And I'm here for any other questions.
Thanks, David. I really like the way you laid that all out for us. And we do have a few questions that we will dive into. Question 1, what testing is best for soluble carbon?
Yeah, that's a great question. So primarily the two methods that I'm aware of that are being implemented on soil currently are what's known as POC, which is an active carbon method that's endorsed by the American Society of Agronomy and the NRCS, as well as the carbon analysis methods that are on the Haney test. Great.
And what levels do you like to see?
On a POC test, you're going to be like 700 plus, I think is the good zone, parts per million. And I'm not as familiar with the Haney test, so I don't know what levels those would be in terms of parts per million, but I know he's got those charts on there that show you if you're in your good zone or not.
To interpret it. Great. How important is it to deal with aluminum in plants?
It's a significant detriment to plant growth in a lot of academic literature. We did a literature review of aluminum, and we have a aluminum stress blog post on our website. If in our reading room on our website, you guys can check that out, where it goes into some of the specific biomolecules disruption functions that happen with aluminum. So generally speaking, yeah, we want as little aluminum as possible, and it's very important. How that happens and why it's necessary and to what degree really varies on a crop by crop basis.
Okay, thanks. What sodium issues are you seeing, especially when base saturation is high?
Yeah, so saline stress and osmotic stress as a crop, abiotic stress influencers and diminished growth regulators are very, very pivotal. We see these in a lot of different soil types, primarily in looser soils and sandier soils. If you have a high sodium base saturation reading, Those are definitely crop limiting indicators that we find on soil tests.
Okay. And
Was there something more to the question, like something deeper there?
not that I know of.
Okay.
Somebody in chat, shout out via chat if there is, and we'll try to get to it. But we do have questions rolling in, so I'm trying to keep up here. What does liquid manure do to soil? And there's a couple of questions in this one. So what does liquid manure do to soil? Injecting versus broadcast? Does it harm microbes? And this is a dairy farmer speaking here.
It's tough to say what the manure is going to do to a specific soil. It can have really great levels of nitrogen and soluble carbon in dairy manure. They can also have salts. They can also have other sorts of chemicals that come in through antibiotics or feed in the manure and the animal waste. But it's going to be really a case by case basis how those animals are managed, and what that individual sample might look like.
Does the injecting versus broadcast influence how you would answer that at all?
There's a lot of we're seeing on the market today a lot of like liquid chicken manure, liquid pig manure, liquid cow manure, that sort of thing. And so I think some of those products I've seen do some really nice things in crop management and crop development. I do think that by the time they're processed, they're somewhat microbially inert. I do think that raw manure definitely has a lot of microbes in it, but whether those microbes are going to be beneficial or non-beneficial or harmful would be dictated by how hot the compost is, what the carbon and nitrogen ratio, aeration, pile turning, some of those other things.
Okay. What advice would you offer for improvement of an alkaline fen in Michigan?
Was that an alkaline what?
Fen, F-E-N.
I'm not familiar with that.
I'll skip that one. I was wondering if that was like a secret soil word. I didn't know. Okay. What SAP test would you recommend for high altitude, 6,500 to 9,000 feet, for perennial grass hay pasture?
We have a whole list of sampling guides on our website, and the grasses, I believe, have their own sampling guide, I believe, up there. They're typically, I think, 4 to 5 times per season in a grass. I don't see any difference in sampling protocols from low altitude to high altitude. I don't think that has really any influence. So primarily you're looking for, generally speaking, 1 quart Ziploc baggie of new leaves and old leaves, harvested in the morning without dew, in separate bags, sealed in Ziplocs, delivered as quick as possible to the lab.
Okay, perfect. You're not done yet. Sorry. What nutrients do you commonly see over applied, and which tend to cause the most damage?
Every nutrient can be an excess. Every nutrient can become an excess. We see, obviously a lot of nitrogen over application. That's a very, very key one. I think the under application of carbon proportionally is arguably the bigger thing to look at. In other words, as the carbon or as the nitrogen gets over applied to one degree or another, you're taking a exponential step away from having carbon synthesis in your plant, which produces all those higher level molecules that everybody wants. So nitrogen definitely, I think the induced excesses, nitrogen and potassium are the typical induced excesses.
Mm-hmm. Okay. Are non-mobile nutrients more difficult to adjust?
Not necessarily, especially it depends on if you have what types of application technology you have access to. So, if you have pivots or, a way to fertigate, drip irrigation, foliar sprays, those sorts of things, you can really adjust really any nutrient relatively easily. I think the bigger question is the material formulation, the other nutrients that are in the plant that may be blocking those nutrients from being intaken by the plant, or some combination of 1 and 2.
Okay, so if you do have non-mobile nutrients and they're at a toxic level, how do you decrease those or deal with them?
Well, so carbon fertilization, as the heart of the new paradigm, really has a way of detoxifying plant nutrient excesses. So that's the key thing. Secondarily, there's going to be synergistic elements that will help that element be intaken more, as well as there will be antagonistic elements that will help to reduce that element's influence. And so, if you apply a combination of carbon as well as synergistic nutrients and antagonistic nutrients that may be in deficiency as well, oftentimes you can depress an excess a little bit faster that way.
Yes, and I do recall a presentation you did earlier this year that went through a lot of those different relationships. It's really cool. So follow
For
up.
sure. Yeah. We're very, very keen on looking at all the interrelationships.
Perfect. Is there a difference between soluble carbon and active carbon tests? And is there a good range for active carbon?
Yeah. So the range for active carbon is basically that you want is above 500 to 1,000. I think 750 is where it gets to you start to get into some good stuff. There is a slight difference in the analysis of those tests. So soluble carbon on a Haney is straight soluble. My understanding of it is it's straight soluble from a water extract. The active carbon on epoxy is accessible generally through plant root development.
Okay, and I have an update on the fen question, which I was not aware of. So the question again was, we'll see if you know still, what advice would you offer for improvement for an alkaline fen in Michigan? And then with the note, a fen is a type of peat accumulating wetland fed by mineral-rich ground or surface water. It's similar to a bog. Any
Gotcha.
thoughts?
So it's a high pH, high organic matter bog. Is that correct?
Sounds like it. Accumulating.
What advice would I have for that system? I would say run a complete soil analysis method on it and tear it apart. Total Digest, Malic 3, Morgan, and water soluble all in one. It's available in our lab, $150. We've analyzed everything from beets to bonsai and through that sort of a protocol, and it really does do a good job if you know how to read it. Of pulling data out of hard to understand soils, whether they be high pH alkaline sand or a peat bog or some sort of a soil media that's been engineered or that sort of thing. But I can't without looking at some tests. It's hard to really say what might be in excess. What I can say is a lot of those high organic matter soils have really high EC. But I just don't know without looking at some greater data. It's just too difficult of a question for me to talk it too blind.
Sure. Sounds like some direct follow-up would be great on that one. That is all we have time for questions, David, but excellent as usual. You covered a whole lot of ground in that amount of time. So thank you.